Okay, welcome everybody. Welcome back to seminars.alife.org. This is our second seminar of the year, and it's a real pleasure to have Sam Kriegman here from the University of Vermont, who's going to be giving today's seminar. Before we get into that, I just want to give a quick reminder about how this all works. So this is a Jitsi-based seminar. You've probably figured out how to join if you're seeing this, but if not, and you're watching the video of this after the fact, then you could have joined live by going to seminars.alife.org and finding the event page and just clicking on the link. You don't need to install any software. You just need a Google Chrome's browser, and everything, fingers crossed, should work from there. One more piece of administrative information is that we are looking for more speakers. We've got 3 lined up over the next few months, but we're trying to fill a schedule till the end of the year and beyond. So if you're interested in giving a talk, then please get in touch. And also we're looking for hosts. If you're interested in doing what I'm doing right now, then please also consider getting in touch. We already have a couple of guest hosts lined up, and I like the idea of keeping it that way, keeping it diverse. So please consider getting in touch. The only other thing to comment on now is if you don't fancy, so at the end of Sam's talk, there's going to be a Q&A, and you can ask questions here within the Jitsi session by unmuting yourself or by raising your hand. There's a raise hand button in the lower left-hand corner. There's also a chat window in the lower left-hand corner that you can open up. But it would be preferable in the recording, people can't see that chat window. So I'd really prefer it if you possibly could to ask questions using your voice rather than the chat. And if you're unable to make it to the live session, then on the seminars.org website, there's a forum type interface where you can ask questions, and I'll be pestering Sam to take a look at those questions and answer them in the days following the seminar. I think that's it from me. And yeah, it's a real pleasure to have Sam here today. Sam's talking about computer-designed organisms. And this is work that I actually did first stumble across in the mainstream media. Sam was just telling me that it was featured in the New York Times, which is pretty awesome. It's really, really neat to have ALIFE style work getting so much attention in the news, and I'm really excited to learn more about what you've done, Sam. So I'm going to turn it over to you. And in an effort to try to kick a bit of a trend off of keeping things a bit informal and fun here, I thought it would be nice to ask you, Sam, what the first ALIFE conference was that you ever went to, and if you have any impressions or memories or anything like that, people you met, anything that the community might be interested in hearing about. And then after that, please take it away and give your presentation. Thank you. Thanks for being here.
Sure. Thank you, Matthew. The first ALIFE conference I went to was 2 years ago, 2018 in Tokyo. And I was at Gecko in Kyoto the week prior. Oh, yeah. And it was 2 weeks in Japan, which was an incredible trip. Best I've ever eaten in my life.
Awesome.
That alone, it was worth it. ALIFE is by far the most intellectually stimulating and interesting conference I've ever been to. That's the first and only ALIFE I've been to. I'll obviously be at the virtual one that University of Vermont, which is where I am now, is hosting.
Yep.
So online, so it's pretty easy for everyone to get to. So my first ALIFE was just incredible. There was a talk by Rod Brooks, who's one of my intellectual heroes. So I enjoyed that. And it was just a fun experience.
Yeah, awesome. Yeah, that was a great conference. My first ALIFE experience was also in Japan. Very similar, amazing combination of work and eating. It was great. Yeah, so great. Thanks. I'm looking forward to your talk.
All right, thank you. So I'm going to share my screen now. Let's see. All right, I've got a second screen so I can make sure that everything's working. And it looks like it is. Okay, so as Matthew said, I'm going to be talking about computer-designed organisms. And I want to thank Matthew. I also want to thank Lisa Soros for helping coordinate this and inviting me to present this ALIFE work. I'm Sam Kriegman. I'm a PhD student hoping to graduate. I'm at least defending in October of this year. This work was done with Douglas Blackiston and Michael Levin, who are at Tufts University, and with Josh Bongard, my Dr. Vader, my PhD advisor. So this image here actually shows a swarm of computer-designed organisms. They're the little beige spheres that are congregating in the lower right-hand corner of your screen. At the moment that this was taken, you can also look back at their behavioral history, the traces that were carved by their movement in a field of dye particles that were evenly displaced along the surface of their petri dish. As they moved in corkscrewing and spiraling patterns, they scooted their body along the bottom of the dish and created these traces that you see here. It's very visually arresting. I can't take too much credit for this because Doug Blackiston is really the one that took the picture and made the organisms here. But it was selected as one of the best science images of the month by Nature. They called these things living robots. This was also somewhat validating 6 months after an earlier version of our manuscript was rejected by Nature. They wanted back in after they saw how cool it was, how cool the final project or the product was. So living robots. What exactly are living robots? Is this a robot, an organism, or is it something else entirely? So here's a video of the organism moving. It doesn't look like it's coming across the stream so well. So let me try one final effort here to just slow down the video.
I don't know about everybody else, but it looked reasonable to me, just so you know.
Oh, it did? Okay, that's great. Well, this is the only video that I just like to slow down because it's my favorite one. And you can see the organism pulsing and moving with its heart muscles. So this is a robot in the sense it was programmed to move forward. And the way that it was programmed was by its design. So its overall shape and its overall body shape and its position of cell types was designed by a computer to perform a specific task that was injected into the computer by a human designer. So in that sense, it's a robot. But like every organism on Earth, this creature is made out of cells, and they heal themselves if damaged. They can't reproduce, but neither can naturally occurring things that we agree are organisms. So in summary, I don't know. That's going to be a reoccurring theme in this talk, is that a lot of it remains mysterious, and that's what's so exciting about this research field. And they were dubbed xenobots, and that's kind of the name that stuck because, come on, it's pretty cool. The reason why they have that name is because the cells that they're made of are harvested from very early frog embryos. The taxonomic name of that frog is Xenopus laevis. Xenopus, Xenopus, laevis, levi, it's potato, potato, however you want to pronounce it. So they're either xenobots or xenobots. Whatever you want to call them is fine with me. This is what that frog, the Xenopus, looks like. And its name actually literally means strange foot, which you can see its little claws, smooth, slippery skin. But this morphology is just one of the possible outcomes that lives within its developing cells. What else can we build with cells? But more generally, and this is kind of the principal reason of ALIFE research existing, we want to understand life as it could be. And nature has only explored an exceedingly small part of the design space of life. What else is out there? I hope that by trying to build new organisms, we might be able to uncover some new designs and things we haven't thought about before. But also, so that's the intellectual pursuits, but there's more pragmatic reasons that you want to build programmable organisms. I believe that they could provide extreme social utility because most technologies now are made out of things like plastics, metals, chemicals, electronics, batteries. These things degrade over time, and they produce deleterious side effects to our ecosystems and to our own health. So the question is, can we build biodegradable machines? Can we build machines that perhaps could even clean up some of the mess left behind by other technologies, but without adding more pollution as they do it? Can we create machines that sequester rather than emit carbon? There's also jobs where current robot technology really can't go. And for that reason, currently there's no way to internally ablate brain tumors. Even if we had a device that was capable of doing that at that small scale, it wouldn't last very long in our body, or it would face a severe challenge of not being rejected by our immune systems. So one of the questions that I think is interesting to ask is, can we build machines that can bypass our immune response and serve as kind of a helper to our immune system? So we could build machines potentially out of our own stem cells that aid our immune systems and continuously patrol our bodies, seeking out little microscopic cancer cells. That obviously is very far off. When you see the things we actually created, you'll know that that is not something we're doing anytime soon, but that is the potential that might be released by creating eco-friendly robots out of biological tissue. Well, what's been done before? Well, there's been human-designed rather than computer-designed, biohybrid machines rather than completely biological machines. One of the early examples of this was heart cells embedded in a silicone jellyfish morphology. And with enough heart cells and with the right shape of the silicone etched out, you get swimming behavior at the centimeter scale here. Later on, some of the same researchers, they transfixed some of these cells so that they were sensitive to light. So there's heart muscles embedded in a stingray shape. And when the light flashes on the left side of the stingray, just like if you were alone in a canoe and you were rowing on the left side of your boat and nothing was happening on the right side, you would turn to the right, and as do these stingrays. So this is swimming. This is at the centimeter scale, externally controllable, and it has this scaffold that thousands of rat heart cells are embedded in. It also has gold in it, just to make it a little bit more flashy and cool. What about designing new organisms? What if we wanted a system, an organism, or robot, or something to simultaneously manipulate thousands of objects at scales from the meter scale to the nano scale? It had to manipulate all of these objects without letting any of them touch. What should such a system look like? It's hard to imagine something like that. Maybe it would be something like Hans Moravec's Bush robot pictured here. But thinking about the kinds of body plans that will help or hinder a new organism performing a new behavior in a new environment. There's no reason to expect that that should look like an existing organism performing some old function in a different environment. But moreover, even if a jellyfish is a great design, but is it the right design for micro scale swimming in a calm lake, the rapids of a river, a tortuous pipe? What about at the scale of a cruise ship? Should it still look like a jellyfish? Not to pick on these things too much, they're phenomenal work, but just to drive home the point a little bit further, is the stingray is a great body shape when you're built out of stingray parts and organs and tissues. But what about when you're made of silicone and you have motors that are rat heart muscles and other kinds of things? Is that still a good shape, or the best shape, the most energy efficient, the easiest to make? One of the few quotes that I'm proud of that are in the press that I said was, what should a robot look like that's crawling through your arteries? Should it have 4 legs? Should it have 2 legs? No legs, 1,000 legs? Should it look like a snake? I don't know. But the very fact that it's difficult for me right now to even name shapes that don't look like organisms that I've seen before, I think highlights the fact that at least I could use automated design to help me be more creative. I can reduce the bias. Instead of making machines that look like familiar objects, I can cede some of the responsibility of design to AI. Even if there's still a human in the loop, I think that AI can be this really creative collaborator to work with because it doesn't share the same biases that we have as humans. So of course, the famous example here, and the reason why I'm even interested in evolutionary robotics, is Carl Sims' Evolved Virtual Creatures. He gave evolution these rigid boxes, and evolution could change their size, their thickness, attach them in different locations by hinge joints, and then evolve them for different tasks like swimming, phototaxis, fighting over resources, walking on the plane, doing whatever that is, all kinds of interesting, non-intuitive designs. And just like all other science, it's not just one person that invented something. There were other people at the time that were doing similar work. There was something in the air, especially at MIT, which is where Carl Sims and Jeffrey Ventrella were. And they were both working on this idea of co-evolving morphology, the body, the structure, and also behavior, and the controllers that were inside of these creatures. They were both interested in computer graphics. And for them, it turns out that for a lot of applications, it's actually easier to simulate physics than to hand design a creature moving by your intuition. So this was useful initially for computer graphics and was adapted later on in robotics. And here are other papers. I'll make my slides available offline. I don't have time to talk about all of these, but I just want to mention the last one on this list by Deepak Pathak and his colleagues. They paradoxically evolved virtual creatures without evolution. I still consider it evolutionary robotics, but they used a reinforcement learning algorithm, and in the action space, it could attach and detach different modules while it was behaving. So this is, I thought, a very creative way to go about evolving virtual creatures. Of course, you could argue that evolution and learning, they're all the same things, just generate and test. But this was certainly novel in my view. I suggest checking out all these papers. I really enjoy them, except for the second to last one. I don't know who that Kriegman guy is, but you can skip that one. So evolving virtual creatures in simulation, that can be challenging, but it's even more challenging to take computational agents and transfer them to physical machines. All kinds of issues in the real world that are difficult to simulate or are just kind of glossed over, because simulation has this finite depth, whereas the real world has, for practical purposes, infinite depth. You go outside, you lift up a rock, there's worms crawling under the rock, you just dissect the worm, you look at it under a microscope, and it has cells, and inside the cells, there's mitochondria. It never ends. There's all kinds of amazing complexity in the real world that's really hard to capture in the simulation. But we can try, and under certain situations, we're able to, in rare cases, transfer behavior from simulation to reality. And this is a paper by Hod Lipson and Jordan Pollack that was also extremely influential to me and everyone else in evolutionary robotics, where they evolved Carl Sims-like creatures and then 3D printed them. They even printed the ball inside the socket joint, which is super cool. And there's some scaffolding that's in there that will be torn apart. And what you'll see right here is a design that was made by the computer, 3D printed, and then just the motors were snapped in and it was powered by this tether. So these, in my view, were the first evolved robots, super inspirational. And this PDF sat in our Google Drive while we were writing our Xenobots computer designed organism paper. This was the model. So they did sim to real. Oops. They did sim to real. We're interested in sim to life. To do this, we proposed a design pipeline, an automated design pipeline. And the input to that pipeline are some building blocks, passive block, contractile block. And we also gave our automated design pipeline a goal to move. And here's what those building blocks look like in pairs. It's easier to see the dynamics of these blocks in pairs because of how it's simulated. Technical details don't matter. You see the red one changing to green as it expands, into red as it contracts. The cyan or blue colored pair of passive voxels just sits there. When we take the individual blocks from these pairs, we put them together in random configurations, it's extremely unlikely that they're going to move. So the cardiac tissue here, the contractile building blocks here are randomly moving with randomly assigned phase offsets. And it's unlikely that a random configuration of how you're setting up those blocks and putting them together is going to produce forward movement. And the design space here is absolutely enormous. So at every position in the design space, you can have either no voxel, a passive voxel or block, and a contractile voxel. So that is 3 to the 448 or 5.63 times 10 to the 213. There's no way that you can exhaustively search this. That's where something like evolution comes in pretty handy. So to evolve something, we need to have some system of inheritance and a genotype to encode this body and pass to the offspring. We use compositional pattern producing networks. So I'm going to go through slides here about the basic understanding of how they work. The input to a compositional pattern producing network is some coordinate frame. Here, X coordinates and Y coordinates, and the output is G, and that's just the amount of gray that is going to be painted in this 2D space below the network. So here we have X fully connected to the amount of gray, and you can see from X equals 0, the whole column is white. And as you move across the image, it becomes increasingly gray until you get to the maximum gray, maximum X coordinate. Do the same thing with Y. If Y is connected to the amount of gray, you see a gradient from white to increasingly gray pixels going down as the Y coordinate increases from 0 to, normally we normalize this between 0 and 1. We can add activation functions in the parlance of neural networks, or interaction functions, perhaps in genotype speak here. We have some kind of threshold where it's all white until it hits that threshold, and the X coordinates are large enough that we get a solid gray area. We can add an extra input here so that we're operating in 3-dimensional space. So now, all the way in the corner of this cube where X, Y, and Z are all 0, we have a perfectly white voxel. Voxel is a 3D version of a pixel. As you traverse this cube down to the other corner where X, Y, and Z are all 1, it's going to be as gray as possible. So you get a gradient diagonally through this cube. These networks very quickly become interesting and more complex as they compose different activation functions together to create patterns that normally have symmetry and repetition with variation in there. The reason why you get these kinds of patterns is due to the shape of the activation functions, but also because the inputs are spatial coordinates. So X, Y coordinates that are 2 pairs of coordinates that are close to each other are going to be more likely to be similar than ones that are far away. This ends up being really useful when you're making body plans for robots. So here we have this kind of canonical CPPN, compositional pattern producing network, that takes this input, the X, Y, Z coordinate of every point vertice in this Cartesian grid. So at every point in this grid, you can have a voxel or not, and that voxel here in this example can be green, cyan, or red. So the CPPN is going to compose patterns together, and at the end, it's going to tell you for every input coordinate whether or not there should be a voxel there and what kind it should be. This comes from Nick Cheney's work in 2013, and we use the same encoding in pretty much all of our evolutionary soft robotics work. So we can evolve designs, but we still don't know which one should be built. Obviously, it makes sense to take the best one at the end of evolution to start with. So we're going to evolve over 1,000 generations. That's the horizontal axis here. Increasingly fit robots. That's the vertical axis. It's the velocity. Each one of these lines represents more or less a lineage of different robots that are improving in their velocity over time. You can look at the paper for more details about what these lines really represent. The important thing is we want the fastest robot at the end of evolution. And it ends up looking something like this. We rerun this process of 1,000 generations with some population size of 50 robots, and we just take the best robot at the end. We do that 100 times to get 100 run champions, the best robot at the end of its independent evolutionary trial. But which one of these should we build? We're not 3D printing these like Hod Lipson and Jordan Pollack. We're still constructing them pretty painstakingly out of cells. We can't build all 100 of them, and we don't necessarily want to. We need to filter these designs somehow. The first way we filter the designs is with something we call the Robustness Filter. And what we do here is we apply random perturbations to the actuation. We basically just let the red cells jiggle about in random ways. And we want to find designs that, regardless of how their muscles are jiggling, they're going to move forward. They're robust or agnostic to the way in which their body parts can move because they're just such a good structure. The shape and the position of where this actuating material is, it doesn't matter how it's moving, it's going to propel the organism in a certain direction to produce a desired behavior. Once we know which ones are the most robust to this noise, we are going to filter a little bit more because it turns out that some designs are easier to build, and I'll talk about the exact build method shortly. But it turns out that designs that are speckled with different material types is very difficult to build. We prefer really contiguous sections of tissue. Most of the designs actually have that. But if they don't, we automatically rule them out. The second thing is that it has to be something that's buildable geometrically. We found that there is a minimal concavity that we can actually sculpt into the real organisms that we're manufacturing of about 100 microns. So we can make little tiny gaps in their body, but no smaller than 100 microns, or the developing tissue just will close that gap up because they're so close in the cell. The cells want to stick together as they're developing. So no matter how much we're scraping and trying to keep that open, if it's under 100 microns, it's going to close automatically. So we call that unstable geometry. Third one is that we would prefer to build designs that are mostly passive for two reasons. One is that passive tissue will last longer. As you'll see shortly, we build these designs out of heart and skin muscle, and the heart muscle eats up a lot more energy than the passive skin muscle. So if we want them to live longer, we want them to be more passive. Also, we want to reserve space for other kinds of sense organs and maybe carrying payloads and different things in future runs from design to deployment. So it'd be nice to save that space for other kinds of things and not rely on it to produce muscle movement. Okay, so we have these kinds of filters, and it turns out a lot of the most robust designs are just going to be knocked out. But there is a design. There are a few designs that pass through these filters. The most robust design to pass through the build filter that we've set up was automatically selected by our pipeline as this design. So this one out of the 100 was the first one that we set out to make. Let's zoom in a little bit. We see it has two stumpy hind legs. They're made of muscle, and as it moves, it's going to lean forward onto its chest and propel itself forward regardless of how its red cells are contracting and expanding. Okay, so we did sim. What about sim to real or sim to life? Here is that organism actually carved out of living tissue. Again, I'll go over the process shortly. But this gives you an idea of how well we can faithfully recapitulate the shape of the evolved organism, the evolved design in vivo, in real life in cells. And here's a comparison of their behavior. The top you have in silico in the simulation. The bottom you have in vivo, built out of the actual cells. And you can see that their movement matches. They're moving forward in the same direction. The viewpoint's a little bit different because we're looking down the microscope at the real organism. You can build other shapes. For example, a torus, some kind of L backwards gamma shape with no legs, just like a right angle. Pyramid, one of my favorites, the biped. This gives you an idea of how well we can kind of reproduce these designs with our current build method. I would say it's pretty close, but it almost doesn't matter. The most important thing is that the behavior transfers, not that the shape transfers. Okay, so this is how they're built. On the left, you can see the scale of these embryos. These are fertilized Xenopus laevis embryos. And right now, Doug Blackiston was injecting them with proteins to alter cell fate and also things that we can light up the different cell types to tell if we put them in the right position once they're fully assembled into organisms. This outer membrane was removed, and then Doug is removing the animal caps, the tops of these embryos, and the stem cells in that position of the embryo that are fated to become certain kinds of cells, in this case, skin. It places them in a media that aids in the disassociation of these cells, but that outer ectodermal layer doesn't disassociate so well, so he had to remove it manually. And one thing just to keep in mind here is that Doug is doing all of this without any depth perception because one of the lenses of the microscope is the camera. So there's going to be very fine-tuned scraping going on here. And Doug is doing this all with one eye closed, which I think is just absolutely amazing because the scale of this, of the sculpted organisms is sub-millimeter level. Right now, this pile is probably about 1 millimeter across. It's going to place about 1,000 of these disassociated cells into little wells, little dimples in the petri dish. I'm going to let them develop for 2 additional days. Here's 100 cells as they're developing, hundreds of cells. They're compacting, they're migrating, they're differentiating into specialized cells. Here's 1,000 cells doing the same, compacting, adhering, forming a sphere because of physics. This is developed tissue. And then what's going to happen here with the sphere is Doug's going to go to work, again, in this case with one eye closed. It's going to take a micro cauterizer that is 13 microns wide across. That's smaller than a human hair. Extremely small, extremely precise work happening here. You have to be a skilled microsurgeon to do this. But if you are, all of the steps are laid out in our paper and our videos of how to reproduce this work. And we hope that by open sourcing our code and the way in which these organisms are built, that more labs around the world will start reproducing our work because it's nothing that a skilled microsurgeon couldn't do. There's no special equipment or anything. Okay, so Doug is sculpting a very rough shape. The shape happens to be a quadruped, a microscopic quadruped. This one was not one of the 100 designs that evolved in this particular instance of evolution because of the particular constraints and things we were asking evolution to do, but a quadrupedal design comes up often in other simulations. And this is kind of just a demonstration of the build process. I want to stick on this video for a little bit more because as you see the organism rotating around, you can get a sense of its shape a little bit better. All of these videos are online on our website. I'll have a link in a few slides. It's cdorgs.github.io for computer-designed organisms. If you just Google xenobots, computer-designed organisms, or my name, Sam Kriegman, you should be able to find all of this, all of these videos, which are somewhat mesmerizing to watch. Especially for myself as a computer scientist, everything that happens in biology almost seems like magic to me. I'm still building up my intuition of what's possible. I certainly did not think that it would be possible to build a quadruped out of cells like Doug is doing right now. So this is absolutely amazing. To get a sense of the kind of complexity that is possible here at the microscopic scale, consider that a tardigrade is about the same size as this quadruped. I don't think that our current build method is suitable for building something like this, but it is possible, and who knows what other kinds of complexity we can achieve at this scale. Also, we could just build them bigger if that's an issue. We just need more cells. Okay, so how do we build these bodies? We saw the video, but let's just go through a little schematic here for completeness. So we inject these fertilized embryos with these tracers so we can see where the different tissues go. We're going to label the ectoderm, or what we presume to develop into ectoderm, green, ectoderm skin. The presumptive cardiac tissue or heart muscle, red, another layer of skin. We have this kind of sandwich or Oreo. You stick together. The tissue will naturally adhere. You let it develop for 2 days. It differentiates, compacts, forms that sphere. Doug carves out by subtraction the design. There are other ways to build these organisms. Doug could spend a month precisely placing every single cell exactly where a computer tells it to go, but that would take way longer than this method of making the kind of organism sandwich. This sandwich method, Doug can make about a handful of these organisms a day. Okay, so here's our favorite designed organism with its big muscly hind legs. It's got dorsal ectoderm, ventral cardiac tissue. So skin on top, muscle on the bottom. And here is with those lineage tracers so we can see really how well Doug placed or carved out this design and has the heart tissue in the right place. I'd say he did a pretty good job on this one. This is just one of the organisms that we transferred and just one of the instances of this particular design that was transferred. We repeated this process multiple times, and we want to look here down the microscope and try to see where they move in the XY plane. So along their dish, starting at the origin 0, 0, we can trace the movement of the in silico, the simulated design, and the in vivo organism, the manufactured creature. What you'll notice, first I apologize, I realize now that these colors are not so conducive to some people that are partially colorblind. They look like the same image. The important thing just to realize here is that they're moving in the same direction. They have the same heading. Some of them are spiraling, and in particular in simulation, they spiral a bit more. It turns out that the muscle cells, it looks like, according to this, they coordinated a little bit more than they were in simulation. We very conservatively assumed that they were going to just be randomly actuating in simulation, and that causes, under some instances of random actuation, spiraling behavior in this design. It's asymmetrical. It's got a little bit, it leans a little bit to the right. Also, they were evolved for 1/6 of the evaluation period. So letting it run a little bit further, we get a little bit more spirals than happened during evolution. But the important message here is that it moves in the same direction, but it's still, and here's that video of the movement of in vivo and in silico. But there still leaves up in the possibility that, well, anything transfers. Any organism you build in this way is going to move. The computer actually doesn't matter. How do you tell if computer design really is influencing the competency of this system at all? The control experiment we came up with was just flipping the design over on its back. 180 degrees rotation around the transverse plane means that you have a turtle on its shell. The shape is completely changed. The tissue distribution is completely changed. Let's see what happens when we trace the behavior of this design in simulation and in reality. And it moves significantly less. We control for false discovery rate. Those details are in the supplemental section, and they're not really important here, except to just mention that statistically, these organisms, it seems very likely that, or at least the data suggest, that their movement and their behavior is due to their design, not to chance. Here's a video of us flipping over the organism. So this is upright. That's 75 times speed for your convenience, so you can actually see it moving. And below it, it's flipped over on its back, and it's just kind of vibrating there. So its function was lost, flipping it over on its back. So I've shown from sim to life. We input building blocks, we give it a goal such as movement, we evolve simulations like Carl Sims, we send it to the real world like Hod Lipson and Jordan Pollack, but in our case built out of cells, so the output is organisms. But what's really important is this loop back, because it turns out the first time that we ran the pipeline forward on the first pass, we assumed optimal muscle coordination. Your muscle cells in the organism, they can sync up however you want, just let evolution figure out the best design. What we ended up with was bounding, galloping gaits that were completely airborne. No part of their body was touching the surface of the dish for almost half of their gait. But the real organisms always kept part of their body in contact with the surface of their dish because they had negative buoyancy. The heart muscles were not that strong or not that coordinated. So we put in constraints here. That is, we simulated the water by very crudely simulating hydrodynamics, just perpendicular forces on the surface voxels of the organism, of the design. We randomized actuation instead of evolving the actuation. And so using random phase offsets rather than evolved phase offsets, this created no temporal coordination, which resulted in behavior that much better matched the dynamics of the real organisms that we built quantitatively. But this process is still very much driven by humans, and one of the goals of future work is to automate this process, to somehow have a camera on the organisms or some kind of way to sense how to automatically adjust and improve the simulation. So we can press play, and the simulation can build itself a more accurate model and encode actually the way that heart cells are expected to coordinate in a given configuration, which right now we don't know. So one of the things that we might want to improve if the behavior isn't matching well is the shape. So in the top row here are 4 different rotations of our transferred organism. In the second row, we've classified every pixel as either being cardiac or skin. And in the final bottom row here, we do the same with the simulated design. And we're going to try to measure how similar the in vivo and in silico designs are using 2D images, which is not the best thing we can do. Ideally, we would build a 3D model of the real built organism, but this is the best we could do at the time to quantify this aspect of transferal. We're going to use something called Hausdorff distance to compare shapes of these 2D projections. Very briefly, the way that this works is for every cardiac pixel in B, second row, we're going to find the closest cardiac pixel in C. So this means pairs. For every pixel, we find the closest pixel in the other one in its corresponding image. We record the Euclidean distance between those two given their coordinates. We do the same for every single cardiac pixel. And then we do this for the skin pixels. Find the closest match between the pair of images. The Hausdorff distance is simply the longest, the furthest away of these close pairs. And we're going to rotate all the images so that they sync up to give the best Hausdorff distance at all the different perspectives. Okay, got to give my dog heartworm medicine. Okay. Of course, there are distortions when projecting 3D objects to 2D. Like I said, this is not ideal. You can look up things like the moon terminator illusion or dolly zoom effect. This is well known. You change the perspective on, for example, a door that's opening, and it no longer looks rectangular. It looks like a trapezoid. So this isn't ideal, but it's the best way that we came up with, just as somehow to quantify the shape match. And we hope to improve this in future work. Apart from locomotion, we also did a number of other behavioral goals, such as object manipulation. We added a yellow brick to the environment and tasked, instead of evolution discovering body shapes that propel themselves forward, to select for body shapes that propel that block forward. And what evolved in some cases, or in fact, in most cases, was a little end effector, a little notch to hold and manipulate that object, keep it in place as it pushes it forward. And here's a little anecdotal in simulation and in reality of object manipulation or block pushing. And there's also object transport. Reason why we thought of this is because one of the designs that evolved a hole through its transverse plane, because this was a good way to reduce hydrodynamic drag. The less surface area you have, if you allow for water to pass through you, then you're going to be a little bit faster at moving through water. So this kind of 3D donut evolved. We looked at that and we immediately thought, hey, why don't we stick an object in there and we can do intelligent drug delivery. So we placed that little block in there and just let it move as normal, and it resulted in object transport. But that wasn't selected for during the evolutionary process. So we ran another round of evolution where we explicitly selected for object transporters. So I want to dwell on this a little bit because I think this is a great example of an unintuitive design that also inspired us to not just make something new, but come up with a completely new objective. So I do think that even if there's humans designing everything and making a lot of the decisions, it's useful to have AI as a collaborator proposing just diverse objectives and different kinds of designs and different kinds of new ideas, free from our kinds of bias. Finally, there's collective behavior, which results from just placing 5 of these organisms amid a field of debris, yellow little hockey pucks here made out of voxels. And we see the same thing in vivo. If we coat the surface of the dish with dye particles, let these organisms move around for a while, and they end up often making piles. So there's debris scattered in their dish, and they tend, by the way that they move and interact with these particles, to aggregate them into neat piles, almost like sheepdog herding sheep. They're herding debris particles or dye particles. Here's a video of their behavior. It's hard to see the debris here in vivo, but what you can see is spinning around. If you watch in silico right about now, you'll see the same behavior predicted in simulation, that these designs will come together, rotate about each other, and form almost a better little sweeper of the debris than the sum of their parts independently. This is really interesting, suggests all kinds of future work, like not just cleaning their dish, but designs attaching together to become larger collectives and building things at small scales together, or building their own bodies to be something larger. And then the last thing actually is just that something that was very surprising to me as a roboticist is that these constructs, these devices heal themselves. In hindsight, it's pretty obvious they're made of tissue, but as something that we're programming to move in a certain way, to have this kind of robustness is just incredible, and was really cool to see. So here is Doug is going to cut this organism open, and then we're going to watch it on time lapse over about 10 minutes, just zipper its body back up. So what's next for these self-healing, self-powered, eco-friendly robots? One thing I'm really interested in is automating the manufacture of these organisms, because right now the computer design is automatic, it's computer designed, but the manufacturer is still human built. We'd like to remove the human from this aspect. Perhaps it will let us scale up to larger and more complex and more organisms. I'd also like to automate the feedback from real to sim. I think this is even more important place to remove human design bias, because the constraints on the simulation are not going to be as simple as randomizing actuation in the future. When we want to actually encode in a model of how the heart cells are coordinating amongst themselves, it's going to be useful to use machine learning to automatically determine these correlations and adjust the simulation. We'd like to complexify the structure and the behavior by adding other kinds of cell types. So not just skin and heart, but also photoreceptors, stomachs maybe, so they can eat, neurons so we can learn about cognitive functions as well as just mobility. We can make cyborgs by including synthetic parts, chimeras by combining different species cells. And what's most interesting to the biologists, Mike Levin and Doug Blackiston, is cracking the bioelectric code. So when the xenopus cells and when all cells are building bodies during morphogenesis, they are signaling to each other and they're coordinating, but we don't know exactly what they're saying. We can see them signaling through calcium, but we don't know exactly what they're saying as they're doing this, as they're behaving, developing, and healing from damage. So one of the things that this pipeline could potentially be used for is to design not organisms that are good engineering tools, that are good at performing some behavior, but are actually scientific tools that can kind of advertise their underlying mechanisms. So maybe there are certain shapes that actually will develop and let you see more easily what the cells are communicating. And so that is one of the thrusts of our future research as well. And just for fun, why not build a golf ball sized xenobot instead of just micro organisms. We'll need a lot more embryos to do that. But in closing, I don't know. And that's what makes this research so exciting, is that we're not just solving puzzles that we presume to know the answer to. We have no idea what's going to come in the future, and we really want to create new collaborations and work with more people and have more people improve various aspects of this pipeline, automating it. Maybe there are things that we're doing that are very crude and not the best way to automatically design novel organisms. So yeah, please reach out to me, email me, tweet at me. I would love to discuss evolutionary robotics and the future of this work. And that is all I have in terms of slides. So I'm going to turn this back on.
Awesome. Thanks, Sam. That was a really fantastic talk. Lots of really interesting material there. And we've got a nice crowd of people here to hopefully kick in and ask some questions. So please, those of you who are interested in asking questions, maybe the way we'll do it is if you just type in the chat window here, just put your name, and then Sam or I can sort of call on you in order to ask your question. And as people are queuing there, maybe I can kick off with a question for you, Sam, if that's all right.
Sure.
So I'm curious a little bit about what you see as being the most obvious constraints on moving this all forward. So I was thinking a little bit about the design space prompted by some of your slides, and thinking about there are all these different things that constrain the types of structures that you're going to be able to investigate, right? Everything from the genotype to phenotype map that you described, right? Your way of specifying the morphology of these things using gradients and so on. Also, the material. So you talked about how when you create these small gaps, they automatically sort of fold in on themselves. And then the search space, like how well the GA works at searching and getting stuck in local optima and so on. There's all these different constraints, including the human ones that you mentioned as well. Which one of those do you see as being the most annoying, the ones that's getting in the way of you really pushing this forward and getting really much more sophisticated robots and so on?
Yeah, I think the most challenging thing is feeding back the constraints from real to sim, because we can simulate whatever we want, but there's no guarantee that we're going to be able to build it. So being able, this is just basically the problem of evolutionary robotics, is how do you transfer computational agents to these physical, useful machines using things.
Live streaming is on.
So that is a fundamental question of evolutionary robotics, and there's all kinds of hypotheses why the complexity is kind of seemingly hit a ceiling. Some people argue that it's because the environment is too simplistic. You're just selecting for locomotion on a flat plane. What do you expect to get? Evolution's lazy, so it's just going to give you the easiest thing that works. Others have argued that it's an encoding or an algorithmic problem. I think it's probably a little bit of all of the above. So the more people we have thinking about this and working on this problem, the better designs we can come up with. And as long as they're constrained in the right way, we should be able to see if they will transfer or not.
That's interesting. I guess I expected a slightly different answer, and that some part of me anyway thought that the materials would be seen as like a major constraint at this point, like the tissues, like you talked about them closing up those gaps and it being hard to manipulate them, and not only to manipulate them into certain shapes, but to keep them in that shape. It's interesting to think about the difference between these amazing things that you've created and like the tardigrade that you showed.
Right.
And it has this very clear form, very functional form that is just, yeah, as you're saying, the miraculous aspect of biology, and that seems a challenge.
Yeah, I think that there's a lot of work that can be put into automating the manufacture and making it more precise. So current bio printers that you load cells into, they don't have that precision of sub-millimeter scale. So right now, Doug Blackiston is more precise than any 3D printer.
Right.
So the goal is to work with our material science colleagues to build technologies that potentially can do this at a finer resolution. But also, it's not just a matter of 3D printing something, because this is a living structure that's moving on its own and closing up gaps. So Doug is constantly scraping these things and kind of nudging morphogenesis in the right direction. So it would be kind of almost a paradigm shift also in the way that we're printing things, because it would be seeding these emergent processes instead of printing a structure that we know what it's going to be like. Or they have shifting things that are printed, but you have target structures and target things they're going to shape shift into. So understanding how to guide these complex systems into desirable outcomes or target morphologies, I mean, that's the $1 million question for sure.
Yeah, cool. Yeah, I mean, I think it's one of the things about this work that that gets me excited is the way it challenges some of our perspectives of, for instance, DNA being a plan or a recipe for an organism, right? You're taking cells that have exactly the same DNA as a frog, and you're turning them into completely different creatures. And it's just emphasizing the evo-devo, the developmental side of things. And it's interesting to think about how some of those messages might work their way into, for instance, how you choose to encode things. So I know that in biological development, there are these gradients that developing organisms respond to, but there are also these growth processes, right, where cells are locally influencing each other and helping each other to form into functional roles. And it'd be interesting, maybe that's another place where, where there's constraints put upon the forms of the things that you're explaining in interesting ways.
Yeah, absolutely. I think there's this really beautiful loop from computer science to biology and back. obviously the computer science, we're doing an evolutionary algorithm. So that's something that was inspired by nature. But there's also ways in which the computer science is influencing the biology. So they're starting to realize that this hardware-software distinction we have in computer science is something that's really useful to think about, that the genome is encoding structural components, the hardware of cells, their structure and the way that they can signal, but then the genome's not encoding this final shape. There's some kind of software running on there. I mean, we showed this. We used something that had one DNA and we built something completely different out of it. So hopefully we can use these as scientific tools to try to dissect that problem a little bit further, try to understand a little bit better this software biology, and that could have implications for the algorithms themselves. So it's this constant feedback loop from biology to computer science. I find really fascinating, and working with biologists is great because they ask all these naive questions that at first I'm like, no, that doesn't make any sense. And then I start questioning what I'm doing, and it creates a lot of inspiring moments, not unlike working with an AI colleague. And I hope that I do something similar with my biologist colleagues as I ask all of my naive questions to them. So this is totally interdisciplinary work.
Yeah,
and we're
awesome.
really trying to figure out what are the areas that these disciplines don't really understand, and how can we help each other.
I love it. Yeah, fantastic. It's one of my favorite things about ALIFE in general is this feedback between different disciplines and different approaches. We do have a couple of questions here in the chat window. So the first one comes from Hiroki Sayama, who asks if you plan to make your simulation model more spherical. And he also asked the typical lifespan of the Xenobots.
Yeah, so there's a lot of stuff that I couldn't pack into this talk. So the lifespan of these, without any further bioengineering, they live about 7 to 10 days. They live off of this protein and fat that's just stored in their cells. They use that up, and then they die. And when they die, you could argue about whether that's life or death, and I think that's one of the interesting things about this research. But when they do disintegrate, they just turn into dead skin cells. And that's a lot nicer to have in the environment than batteries and motors. So then the question about making them more spherical or less spherical?
I'm guessing that he's getting at the fact that you've got cubes, the voxels are cubical as opposed to spheres. Is that, Hiroki, if you're here, you're welcome to unmute yourself and ask. Okay, go for it.
Yeah, fantastic work. So my question is that, when you are designing by using evolution computation, you have cubic possibility space. Have you tried a more spherical shape from the beginning, like applying CPPN to the spherical domain, so that the design can be more compatible with what Doug is, working on?
Yeah, absolutely. So right now, for this paper, we were stuck with a CPU simulator. So our resolution at which we could simulate these designs was pretty coarse-grained. We've since moved to a GPU simulator that we can simulate more voxels, which gives you rounder designs, but they still are voxels and boxy. But actually, the way that they are interacting with their environment on the surface is a spherical collision envelope. So there is more smoothness than meets the eye. I could actually just draw these, render them differently in simulation so they look smoother. But it's being simulated by these discrete blocks. It helps me think about the design space a little bit better when I see them as blocks. Then you bring up, it's a really interesting point of whether or not higher or lower resolution is going to yield more transferable designs. So it could be that this coarse-grained simulation prevented us from overfitting in simulation. I don't know, and we're going to have to investigate that further. So if I did a cell-to-cell representation, so there's about 5,000 cells in that cardiac skin bot, but there's only about 300 voxels in its simulated design. So as that gets scaled up to be more to one-to-one mapping, am I going to have to simulate the cells at a higher fidelity? Maybe if I have this GPU simulator, I can simulate a single cell with multiple voxels. So there's all kinds of computational questions there that I don't know the answer to, but what's going to drive that is going to be the behavior. So if the behavior matches, then we're good. If it doesn't, then maybe we need to recapitulate the shape better. But it doesn't matter if you get the shape perfect and the behavior doesn't matter, or doesn't match, then you just have kind of a statue. So we're making robots, so we have to make sure the behavior matches first. And if it doesn't, then maybe we need to increase the resolution or smoothness of the design. But thanks, that was a good question.
Yeah, thank you very much.
We also have a question here from Carlos. Carlos, if it's easy for you to unmute yourself, I'd love it if you'd ask the question yourself.
Yep. Thanks for very interesting work, Sam. I was wondering how easy was to pass the ethics board, because, I mean, when working with lab animals, I mean, it passes. I mean, you just need to justify that you're going to kill them for some purpose. But I don't know whether you face some obstacle, because on the one hand, it's unclear whether the xenobots are alive or not and how you should treat them ethically or not. And also, I mean, starting from the frog embryos. So I don't know whether that was easy or it was standard procedure.
Yeah, so right now they're treated as frogs, which are a lot more complex than agglomerations of skin and heart. So I think they're on pretty good ethical standing right now. There's really strict ethical, as you know, considerations for animals. You can do things in your own home that you couldn't even dream of in scientific studies. Like you can go fishing and kill a mouse in your basement. There's a lot of barriers to doing scientific research with living organisms, and there should be. I do think that even though we're at a really early stage here, who knows where this is going to go? And talking about collaborations, I would love to collaborate or just have philosophers and people working on this that are trained to think about these issues. So as a computer scientist and roboticist, I might not be the best one to think about the ways in which this technology can be used and abused. So we're definitely going to need to have more collaborators thinking about the ethical issues as this technology scales. And I can see, at least from my perspective, there being a couple of issues. There's one, which is a concern for the treatment of the frogs themselves. They live very nice lives. They live a lot longer in the lab settings than they do in the wild. They also, the embryos that we take these cells from, we only use some of the cells, and they're super early embryos. There's no neurons in there. They're just undifferentiated stem cells. And then in terms of concern for the technology getting out of hand, I think that there's all kinds of technologies that are way more dangerous than xenobots right now, and things that can self-reproduce and that are deadly that I think we need to take care of before we get to xenobots. But it's still interesting to have the discussion, I think, in this early phase. We might as well figure that out now.
I also wanted to add, in January, I was in DC at a NSF workshop, and Chris Adami was there, and your paper was just out, so we were commenting on it. And I thought, well, if we consider the cells that xenobots are made of alive, then this should be alive as well, living robots. And then a student from Georgia Tech told, well, I call them, they're, instead of not so much living robots as dying robots, because let's say they won't survive for long. But then you could say the same about any living system, that we are just in the process of dying sooner rather than later. So I don't know whether what has been the reaction of different people towards the life label on these creatures.
Yeah, I think it's a really cool tool to think about. It really stretches our definitions of life and robots and other things, which is fun. Yeah, so from a robotics standpoint, this self-limiting lifespan is a good thing. In case something goes wrong, we know they're just going to disintegrate in a week, so we don't really have to worry about it too much. Of course, there's always a possibility they could evolve in the environment, and we have to be careful about that. And they're certainly not being deployed anytime soon. I think that if you watch the videos and you see these things and they literally have a heartbeat and they're moving, it kind of triggers our imagination. Maybe this is with all biological research, especially if you're using cells or there's development or self-reproduction or anything like that, we automatically start thinking about stories like Frankenstein and Brave New World and all kinds of things. So it certainly triggered the imagination of a lot of people. Most of it has been positive feedback. People are excited. They see that they're very simple, that this is interesting intellectual pursuit. Maybe they could be useful machines and clean up microplastics in the ocean one day. That's kind of far off. Of course, there's been conspiracy theories as well, just like anything else. I try not to watch them, but I can't look away. So I've seen all of those as well. And yeah, I mean, I guess you're always going to get that. And anytime you're working with cells, I think. People didn't really care when all I was doing was virtual creatures. Are those alive? I don't know. Maybe there's groups of people that don't want me deleting my simulations either. I'm not sure. Those seem much more complex than just the lumps of cells moving around.
Great. Thanks for your question, Carlos. We've got one last question here in the chat window from Emily. Emily, are you around to ask your question verbally?
Hi, yeah. So this was a follow-up question to Hiroki's. You mentioned that the reason that they would die in 7 to 10 days normally is that they kind of run out of nutrients. Is there a possibility of feeding them so that they would not die at that point?
Yeah, absolutely. So we can actually put them in a nutrient-rich solution now and let them live months. There could be bioengineering in which we give them stomachs to actually seek out fuel in their environment if that's something we're interested in. I don't think that's off the table. Again, though, I do think that the self-limiting lifespan might be a good thing, but it depends on the application or what we're trying to study. But to answer your question, yes, there's already a way to prolong their life.
Do they ever reproduce or, divide? And
so they do not. They're basically just like if skin cells were shedded off of your own body, or like frogs, they naturally shed. They're just skin cells, so they just die and they don't reproduce. That said, you could imagine building things out of other kinds of organisms that do just reproduce by being torn in half, like planarian, flatworm, or hydra or something.
I just meant the individual cells. The individual cells, they don't divide. They don't go through mitosis.
Yeah, so in the development process here, there might be some cell division, but mostly it's just compaction, migrating, adhering, and differentiation right now. Yep. Cool. They could be bioengineered to do other things. And one of the other lines of research here is, instead of having these cells in a sphere that aren't dividing and then we sculpt out by subtraction, we just guide the morphogenesis of a single dividing cell. And this is active research that we're doing now in the Levin lab. We're trying to think of ways in which the computer can design these kinds of things. It's much more difficult. It's easy to come up with target morphologies to sculpt out of a hunk of clay than it is to figure out how to nudge morphogenesis in the right directions, because we really have no clue there, again, about the software of biology. So
yeah, very interesting.
Thank you.
That sounds like a really interesting avenue for future research. I'm reminded of Lindenmayer systems and so on. And yeah, lots of cool stuff there. And great talk. Thank you so much for taking the time. And thank you to all of those of you that joined and participated in the Q&A. I think we're going to wrap it up there. So thanks again, everybody, and see you next time.
All right. Thank you.